Hello, I'm Curren from Brookland High School. I'm happy to present at Created AState as a YES Scholar conducted by the Arkansas Biosciences Institute at Arkansas State University. I work under Mr. Sankalpa Chakraborty in the laboratory of Dr. Viswanathan Rajagopalan. Today I'm going to discuss our exploration of epigenetics and the regulation of cardiovascular diseases. I would like to start off by giving insight on epigenetics. Epigenetics is the study of heritable changes that do not alter genetic sequences of deoxyribonucleic acid, also referred to as DNA. Several epigenetic processes exist, but this experiment focuses on histone acetylation due to its possible link to CVD. To better understand this, we will explore what histones and histone acetylations are. Histones are proteins that act as a spool which DNA strands wrap around due to an attraction between the two as they have opposite electrical charges. Histone acetylation functions to alter this charge with the addition of an acetyl group to the amino acid lysine at one end of the histone molecule, causing the DNA to unravel from the histone. This exposes more docking sites for regulatory proteins, causing genes to undergo or halt the transcription of ribonucleic acid, also known as RNA. This experiment attempts to discover a relation between histone acetylation and CVD. CVDs are the number one killers of humans, not only in Arkansas but worldwide, and recent studies have shown a link between epigenetics and CVD. Exploring histone acetylation's relation to CVD could lead to better treatment strategies. To determine a link between the two, heart tissue samples are taken from three separate groups of rats are examined. These three groups include a control group, a thyroid hormone abnormality (CVD) group, and a CVD group treated with thyroid hormones. Thyroid hormones are important in this experiment because thyroid hormonal abnormalities can cause heart disorders. These tissues were examined using electrophoresis and Western blotting methods, which I will explain shortly, to determine levels of histone acetylation in each group. The first process is subcellular fractionation, which is the purification of homogenates or liquid samples containing all cellular organelles, using a centrifuge, which is a machine that separates homogenates using centrifugal force revolutions over a given period of time. We use this to target nuclear proteins in our heart tissue samples. The next step begins SDS-PAGE, which abbreviates sodium dodecyl sulfate polyacrylamide gel electrophoresis. Using the detergent sodium dodecyl sulfate, we straighten the folded protein so separation is by size and molecular mass only. Next, we conducted polyacrylamide gel electrophoresis, which uses the positive charge to pull negatively charged proteins through the gel, allowing proteins to be measured. The semi-dry Western blot process is next. This process transfers proteins from the gel to a PVDF or polyvinylidene fluoride membrane. We will do this because the gel is not a suitable platform for antibodies to bind with proteins, but the PVDF membrane is. The gel and PVDF membrane will be placed adjoining one another between two filter papers which have been soaked in a transfer buffer, and placed in a semi-dry transfer system which uses a positive electrical charge to attract proteins from the gel into the PVDF membrane. Next, we will wash the membrane in a blocking buffer called bovine serum albumin, or BSA. This washing fills all empty binding surfaces, preventing the nonspecific binding of antibodies. This means these antibodies only bind to the targeted protein, providing accuracy. We will then wash the PVDF membrane in primary and secondary antibodies, which are commercially available rabbit antibodies raised against the target H3 acetylation proteins in rats. The PVDF membrane is washed in primary antibodies which bind to our target proteins, and then secondary antibodies which bind to each primary antibody and are detectable when developed in an imaging system because they were incubated in a dye and contain an enzyme or fluorophore, which is a light-emitting chemical. After this, the PVDF membrane will be washed in a developing reagent which will react with the dye and secondary antibodies to emit light, which is detectable when the PVDF membrane is developed in an imaging system, allowing us to see the concentration of H3 acetylations. We expect there to be a significant difference in the amount of H3 acetylation status among control and experimental groups. As described in this poster, exploring the status of H3 acetylation in cardiovascular disease can shed light on better understanding how to treat the disease. I thank the opportunity to present in this prestigious symposium, and I aspire to discover major new findings towards better diagnosis and treatment of these lethal disorders. So far, I have been able to perform serial dilution of test albumin proteins, run SDS-PAGE, stain with Kumasi, and destain it subsequently. As soon as the COVID-19 pandemic ends, I look forward to continuing the rest of this process.